a center of knowledge, innovation, history, culture, and biodiversity. This is the University of the Philippines, Los Baños. UPLB found its home at the foot of the legendary Mount Makiling in Los Baños, Laguna, a short distance from Laguna de Bay, the country's largest lake. It has its beginnings in 1909 as the UP College of Agriculture, a tent school for a handful of students. A year after, the Forest School, or what was to become the College of Forestry, also set up its home in Los Baños. In 1972, the UP College of Agriculture and the College of Forestry, that had by then become renowned institutions, were granted full autonomy as the University of the Philippines, Los Baños. Instruction, research, and extension in agriculture and forestry in the country took root and flourished in UPLB. Through its strength in these two fields, UPLB established niches in various areas: biotechnology, engineering, veterinary medicine, natural resources conservation, environmental science and management, human ecology, food science, economics and management, public affairs, and development communication. It drew its strength in the arts and other fields of science, from harnessing its expertise and human resource in the biological, physical, and social sciences. Today, UPLB degree programs continue to dominate over its counterparts. Over the years, UPLB has developed graduate programs that continue to attract students far and wide, including collaborative programs with international universities in food security, biodiversity conservation, and development. To make graduate programs more accessible to professionals, UPLB offers them off-campus. Recently, UPLB established the UP Professional School for Agriculture and the Environment in Mindanao, the country's food basket, in order to help build the knowledge capital that the country needs to attain food security. Instruction in UPLB is enhanced by a strong and vigorous engagement in research and public service that enables it to produce knowledge and generate technologies and innovations that promote sustainable living, making it a strong force in the war against hunger and poverty in the country. Extension and public service are the mechanisms by which UPLB brings this knowledge and technologies to the people, as well as for it to know what they need and to help shape policies in the country. UPLB's reputation as a graduate and research university was formed through the continuous aspiration for excellence and the mutual reinforcement between instruction, research, and public service and extension. It brings in partnerships with local and international universities and provides mobility to faculty members, researchers and scientists, and students, and enhances their competitiveness. Instruction, research, and extension and public service in UPLB reflect the dynamism, honor, and excellence that its forebears set as a standard for its leaders to aspire for.
The world is in a constant state of change now more than ever. It's a world that requires resilience, flexibility, and innovation to overcome challenges and to pursue growth. That is why the University of the Philippines Los Baños is pursuing future-proofing initiatives to provide agile leadership and develop local and global communities amidst shocks and disruptions in an ever-changing world. Guided by compassion, an openness to working together, and the daring to seek out breakthroughs, UPLB constantly pushes for academic excellence, produces relevant research, establishes academic and industry partnerships, and cultivates creativity, critical thinking, and innovation. To keep up with the fast-changing world and to strive for inclusivity in development, UPLB seeks to level the playing field and expand avenues for acquiring new knowledge. It is reshaping the learning experience, enabling students and educators to continue with the learning process in vastly different learning conditions. It is expanding its academic programs and supporting initiatives to create a conducive learning environment. To address the growing complexities of modern problems, UPLB continues to break barriers across disciplines to produce cutting-edge innovations and solutions that serve the needs of various communities. It is taking on challenges in food security and sustainability and coordinating research efforts to direct the university's resources toward critical research areas, making sure that the university-generated knowledge can reach its intended stakeholders and be applied to real-life contexts. UPLB is dedicated to its commitment to contribute to public good and social welfare as it continues to work with the local government and the community to address their most pressing needs. Amidst its engagements, UPLB remains as one of the top regional and global universities and continues to be recognized in the country and across the world. As the national university, UPLB aims to be a steadfast partner in development, as a producer of new approaches that minimize the cost and impacts of future shocks, and by honing leaders committed to knowledge creation, innovation and cutting-edge research, and public service. This is UPLB's roadmap to be an impactful institution in a constantly changing landscape. Responsive, innovative, future-proof.
Okay. So, blessed Tuesday morning, everyone. So, good morning po sa ating lahat. Today is December 6, 2022, and hashtag Team LRC is back for a new set of learning sessions through our tutorial webinar series entitled GENE-ius Talaga, a tutorial webinar series on genetics. So, kumusta po ang bawat isa sa atin? Na-miss nyo ba po kami? Dahil kami po ay na-miss po namin kayo. So medyo matagal-tagal po tayong hindi nakita-kita po sa ating mga webinars. So long time no see, pero we are now on the first week of December or second week na nga, at ramdam na po natin ang lamig at diwa ng Pasko. So kumusta naman po kayong lahat dito sa Zoom at sa Facebook Live? So we're present in both Zoom and Facebook Live. So sa mga nakatutok sa ating FB Live dyan, magandang umaga po sa inyo. Hello kay Maria Cheryl Diendo. Good morning po sa inyo. Dito, we have Sir Angelo Noel Manuel, one of our loyal viewers from NTC. So, taga saan po yung iba natin mga early birds dito? Comment nyo naman po dito sa ating chat box. Dahil tayo lang din naman po magkasama ngayong umagang ito sa ating learning session. Taga saan po yung mga iba dito? Let's see. So actually based dun sa demographics no ng ating 293 registered participants ay makita na maraming mga estudyante across different constituent universities of the UP system as well as other esteemed institutions buong Pilipinas who are very much willing to learn more about genetics. So good morning kay Sir Arjun Calyada from Sintang Paaralan. So good morning po sa ating lahat. From UST, J. Luis Infante. Wow, we have someone from UST. All right. So kung wala pa po dito, yung mga friendships, classmates, and colleagues, so itag na po natin sila dito sa ating FB Live or isend nyo po itong Zoom meeting details at i-share nyo rin pong ating video para marami po tayong matututo sa ating mga mapag-uusapan o matatalakay ngayong umagang ito. Okay? So I hope on board na po tayong lahat. Time is now tutorial webinar series on genetics. So genetics is a subject is challenges students no from high school to sometimes sa elementary pa na may pre-discussion ng genetics until college syempre. So which is why for this tutorial webinar series our team prepared 3 sessions. So compress na natin lahat ng discussion us to fully appreciate the different fundamental based lessons sa genetics. And for our students here in UP Los Baños who are taking Bio 30, which is offered as a core subject under the BS Biology program and a prerequisite to higher biology courses. So we want to serve this as a review. Since this is part of the center's team learning sessions, we aim to provide these learning or upcoming examinations and other assessments. Na? So I'd like to introduce myself. Although I think kilala na po ako ng iba dito. No? So I am Joshua Michael Jonas, University Research Associate and Tutorials Coordinator of the UPLB Learning Resource Center. And yours truly po will be our sessions moderator for this morning. So we are now on our first session on chromosome. So ang awesome ng ating mga chromosomes, no? So we will be discussing about heredity and chromosomal principles. So isa to sa mga unang topics na dinidiscuss usually sa mga modules on genetics. So we will be learning about the basics of genetics as the science of heredity and variation and the chromosomal basis of heredity. No? So magkadigtong yan yung chromosomes at ang heredity. So just a few friendly reminders for our tutorial session this morning. Number 1, please prepare a pen and paper and take down notes and actively participate in the discussion. Number 2, keep your microphones on mute and cameras off unless instructed to avoid distracting the speaker and your fellow participants. You'll be given the chance later. Number 3, use this responsibly. Lagi namin itong sinasabi, please keep your comments helpful and respectful and considerate to the speaker, moderator, and to your fellow participants. Number 4, if you have questions, feel free to write them down our comment section or chat box and make sure that they are relevant to the topic this morning. And lastly, please do not forget to answer the evaluation form. After the session, our team will deeply appreciate your comments and suggestions. So please be reminded that the deadline of responses will be until 10 p.m. tonight. So for those who will not be able to join us today because you have equally important matters to attend to, perhaps you have another class, don't you worry. You can always watch the replay of our sessions on our Facebook page. So we now have 106,000 strong followers. So kami po ay lubusan pong nagpapasalamat sa inyo pong suporta sa aming pong team sa #Team LRC. And likewise, you can also watch the recorded sessions on our YouTube channel. So just search UPLB Learning Resource Center on YouTube. So we now have around 3.38 thousand subscribers. So please continue to like, share, and subscribe. And also click the notification bell to keep you posted with our recent videos. So we would also like to invite you to follow and connect with us on Twitter, TikTok, and LinkedIn. Kindly use our official hashtag for this tutorial series, which is hashtag GENE-ius ka talaga, para mabilis po nating makita ang lahat ng videos ng ating tutorial webinar series using that hashtag together with hashtag Team LRC na rin po. Okay? So, moving on, I know it's just 9:07 in the morning. So, tayo ay mag-energizer muna. So, palagi po natin itong ginagawa. Webinars or the drill, hindi na kailangan sabihin yan. So, first, open a new browser or a new window and kindly go to www.menti.com. And second, kindly use the key code that you see on your screen, which is 64294133. Tapos may mga dalawang tanong akong nilagay dun sa ating Mentimeter. No? Okay? So, allow me to share my screen. Ayan. So, first question natin. What comes into your mind when you hear the word genetics? Ano nga ba ang naiisip niyo pag narinig yung subject na genetics, no? When you first encountered it? So, we have here study of genes, genetic variation. Gene, syempre, no? Kasi nasa word nga na genetics. Inheritance, DNA. Tanda pa ba natin kung ano ibig sabihin ng DNA, no? Genetic engineering, pedigree, inheritance, ano pa ba? Try natin ilan ba tayo dito sa Zoom? We are 32 here. Let's try natin maka-15 perhaps. And kasama din natin yung mga nasa Facebook Live, no? Phenotypes, phenotypes, genotypes, magkadikit yan, na? Ano pa ba? Heredity, gene, DNA. Wala pang nagsasagot ng RNA, no? Phenotype, pedigree, genotype, phenotype. Yan, heredity, lumaking heredity. 5 pa. Let's look for 5 more respondents. Ano natatandaan ko sa genetics ko nung high school? Hindi ko matandaan, Sir M, kasi hindi ako hindi ako nag-Bio 30 nung undergrad ko sa UPLB. So, good morning po kay Sir Chico nanonood po sa Facebook Live. Hi, sir. Good morning po. Ano pa ba? Sige. I think nagpapainit pa yung iba nating mga kasama. Ay, Gregor Mendel. Oh, sino kaya yung Gregor Mendel na yan? Medyo kilala ko yan. Lagot ako kay Sir Eman kapag hindi ko kilala yan si Gregor Mendel. Tapos ano ba yung experiment niya? Ano ba yung ginamit niya experiment? Yung kanya mga teapods. So, di ba? Yes, may natatandaan pa ako kahit pa paano. Siguro, let's proceed to the next question na. No? So, we have here 12 respondents. So, these are good answers. No? And usually, the common answers that comes into our mind when we hear the word genetics. Genetic variation yan. Right? Second question. What do you want to learn more about regarding yung topic natin, chromosomal principles? Ano pa ba ano ba yung mga, para lang ma-idea si Sir Emma, ano ba yung gusto nating matutunan sa topic natin on heredity and chromosomal principles? Siguro I'll wait for 3 responses siguro. Naputol ata yung question ko dito. Pero what do you want to learn more about heredity and chromosomal principles, which is the topic that we have for this morning? Ano ba? Ako Sir Eman, personally curious ako kung how consistent ba yung ating laws on heredity? Is nabe-break ba siya or talagang hindi talaga siya matitibag, no, yung ating sa laws on heredity? Or ano ba yung mga iba pa mga components sa ating mga chromosomes? Variation or mutation? Yes. Ano pa? Siguro I'll wait for one more answer. Although ito yung mga madi-discuss naman ni Sir Emma. Ano pa ba? I'll wait for one more. Ano pa ba? Mutation, heredity, yung yung kasama ata, yan, the Punnett square. Yes, yung ating mga dominant and recessive allele. Chromosomal behavior. Okay? So siguro that's all for our energizer, and at least may idea na si Sir Emma no sa mga benchmark knowledge na ng ating mga audience or participants this morning. Okay? So, hindi ko na papapatagalin pa. Let's now go to the main part of our session. So, our speaker, our distinguished speaker for this morning is an assistant professor at the Genetics and Molecular Biology Division of the Institute of Biological Sciences, College of Arts and Sciences here at the University of the Philippines, Los Baños. Here to share with us valuable insights on heredity and chromosomal principles, please welcome in the Zoom space Assistant Professor Emmanuel T. Galang. Good morning, Sir Eman.
Morning, Sir Josh, and the rest of the LRC team. Good morning din mga participants. Hi.
Good morning, sir. I think ready na po ang ating mga participants.
Yes po. Shall I take it away?
Yes po, Sir Eman. Thank you.
Alright. Sige, so prior to starting, mag-share screen muna ako because normally when I do my lectures, nag-chalk and talk ako. So eto na siya. Best ko na to. One sec. By a cable. Alright. So, ayan. Kita ba yung screen ko, guys? Kita siya? There. When we speak of genetics, we are generally looking at chromosomal behaviors to begin with. But actually, I'm supposed to start my topic with this, but I was somehow inspired with the way you answered the Mentimeter a while ago na anong gusto nyong aralin sa genetics or bakit or whenever the term genetics come to mind. Whenever you hear the word genetics, what comes to mind? Ganon. Genetics is one of the most interesting fields in biology, if not the most interesting fields in biology. I remember when I was taking my comprehensive exam during my master's, while I was taking my master's degree in UPLB. And then that question was popped during my comprehensive exam. And then the question was, why study genetics? My answer to that question was, somehow dala din ng kaba kasi comprehensive exam yun. Parang, I said, when you study genetics, you study the past, the present, and the future. In the sense that when you study genetics, you study the past. Dito muna ako magsusulat, i-erase ko lang to. Whenever you study genetics, you study the past. Because whatever you have right now, it is a product of the passing down of the chromosomes from one generation to the next. So in short, the chromosomes in every cell of my body connects me to my past because of the fact na ito siya, yung the same chromosomes na kinary ng mga ninuno ko. And also, we study evolutionary processes and other historical events that are key to the process of evolution to what we are now as a society nowadays by looking at the genetic sequence. So there, whenever you study genetics, you study the past. At the same time, whenever you study genetics, you study the present. Hello, sir. Present genetics? Yes, the answer to that question is yes. You are studying genetics. You're studying the present whenever you study genetics. Because at this point in your lives right now, every single cell in your body is undergoing the processes that are necessary for us to interpret the genetic material. So this includes the central dogma of molecular biology, wherein the DNA in your chromosomes are being read, transcribed, and translated at this very moment to produce the necessary products that your cell needs as of now. Say, for example, I just had my breakfast. So therefore, my cells, the individual cells in my body will respond to me eating breakfast by expressing the genes that are present as DNA in my chromosomes so that it can produce whatever I have ingested, which is happening in real time at the present. By studying genetics, I study the processes that are currently happening inside my body. Also, sir, pati future ba naman? Ano yung genetics manghuhula? The answer to that question is yes. The future to all advancements in biology will always have a genetic component. Say, for example, probably you've heard of genetically modified organisms. Yes. So that is the future. At the same time, if you have encountered the term CRISPR, or which is one of our most modern genome editing tool. So in short, the future directions of medicine, of agriculture, of animal science and breeding, of let's say of agronomy, so on and so forth, will always be genetic in nature. Because once we understand the blueprint that discusses life processes, phenotypes, and other things related to life, we can know more how to manipulate it to further help our society as a whole. So in studying genetics, you study the past, the present, and the future. And in studying these three, we start with chromosomal behavior, of course. Because your chromosomes are, if we will be asked this question, what are chromosomes? Yung sagot lang dyan is very simple. These are carriers of genetic information. There. So your chromosomes are carriers of genetic information. And if we will take a look at mainstream media portraying chromosomes, ito yung palagi nating yung Days of Future Past, makikita natin yung karyotype ni Mystique. And it shows all of the chromosomes ni Mystique in that particular movie. And we all observe that they all look like this. And eventually, these structures are generally rod-shaped in nature and are commonly found as DNA. So these are long stretches of DNA that are packaged in such a way na mas madali siyang i-handle ng cell. So this involves long stretches of the oxyribonucleic acid packaged very well using DNA packaging proteins such as histones and non-histone proteins, so that the long flimsy stretch of DNA becomes more stable, more solid, more rigid as chromosomes. So regardless of the appearance of the chromosomes, because later we will see na maraming itsura yung chromosomes, they will always contain similar parts. So what are these similar parts? These similar parts include, number one, this is the arrow points to the centromere. Ayan siya. And this is referred to as the primary constriction. Ito guys, sa mga kukuha ng exam, regardless if licensure exam pa yan or Bio 30 exam in Bio 30, whenever you're asked kung gaano kadami yung chromosome sa isang organism, always count the centromere. Your centromeres will never betray you. Okay, so the centromere is referred to as the primary constriction, and this is where kinetochores are generally found. So the centromere itself is not a structural feature, but instead this is more kinetochores are generally found, and these play a huge importance in the process of chromosomal segregation and chromosomal movement during mitosis and meiosis. There, so that's the centromere. And also, a huge chunk of your chromosomes are referred to as your arms. So this is one arm, and this is another arm. So generally, there are chromosomes with varying lengths of chromosomal arms, like this one in this diagram, wherein we indicate the shorter arm as the P arm and the longer arm as the Q arm. So P stands for petite and Q stands for Q, spelled as Q-U-E-U-E, which refers to a very long line. So ayan siya. So P arm and Q arm. So in these arms are loci. So eto, say for example, a locus is a definite position in the chromosome arms that codes for a particular gene. So say for example, in this locus, you will find the instruction for plant leaf color. Or say for example, in this locus, over here, you will find the instructions for, let's say, the height of the plant. In short, in these rod-shaped structures, the genetic information is generally confined in the arms, whereas the centromere will only play structural roles in chromosomal movement. And lastly, ends of the chromosomes, as shown here, colored in blue. So these are referred to as your telomeres. So your telomeres play a very important function because since located sila sa ends ng chromosome natin, they have this role that is protective in nature. So yung kanilang trabaho is to protect the chromosomes so that these do not get degraded in the presence of harmful enzymes. So these protect the chromosomes, and on top of that, this will prevent the chromosomes from sticking to one another at the specific ends. So that's the function of your telomeres. So kung titingnan natin, a centromere is the primary constriction. The arms make up the larger bulk of the chromosome, and you will find locus of a gene. Plural of that is loci. Kung marami, so say for example, you have one locus here, you have two loci, therefore, so loci na siya, plural. And also, on top of that, you also have telomeres. And your telomeres play a protective role in the chromosome. So lahat-lahat ng pinapamana from one generation to the next, the information that is necessary for life are all confined in this very small structure. But is visible under the microscope. So this is your chromosome. Okay. Probably you have encountered the terms the term chromosome number. Ano ibig sabihin ng chromosome number? Say for example, when flashed this particular figure, this is actually a valid chromosome number. And you can see that this is a two-sided equation. Two-sided siya in the sense na meron siyang left side and meron siyang right side. Very simple. The left side tells you of the ploidy. Or the ploidy indicates the number of sources of the chromosome. So therefore, in this case, 2n siya. So since 2, therefore, the ploidy level is 2. Therefore, this is a diploid, as we can see. So 2n. Okay, so the first part of the equation, the left part tells you of the ploidy level. So this is a 2n. So therefore, this is if 3n siya, or a tetraploid if 4n, or a pentaploid if 5n. But for starters, let's start with a diploid. So a diploid indicates that you have two parents because the number of sources also indicate the number of parents. As humans, since meron tayong chromosomal material coming from the mother, so that's one source, and chromosomes coming from the father, that's another source. So we are also diploids in nature. So may 2n din tayo sa ating chromosome number. Okay, tingin tayo sa right side of the equation, 4. 4 simply tells you of the number of chromosomes you will expect at metaphase. So eto, sinasabi lang sa'yo na there are 4 chromosomes. So there is a total of 4 chromosomes in this particular example. So ayun, so 2n equals 4. So 2 sets making up 4 chromosomes. So therefore, kung 2 sets to siya, so therefore there are 2 specific types of chromosomes in this example. So this is chromosome, say for example, this you have chromosome 1 coming from the father. So 1 paternal, and chromosome 2 coming from the father. And at the same time, you have chromosome 1 coming from the mother, and chromosome 2 coming from the mother. So kung bibilangin natin, ano nga yung divine rule natin kanina? How do we count the number of chromosomes? We count the centromeres. 1, 2, 3, and 4. So it reflects that you have a total of 4 chromosomes. And then how many sets do we see? we have a maternal set, and at the same time, we have a paternal set of chromosomes. Therefore, making sense to the 2n at the left side of the equation. Okay? So 2n equals 4 tells us that there are 4 chromosomes, and there are 2 sets of chromosome, one that is paternal and one that is maternal. Okay, so practice tayo. What if you have, dagdagan natin ng isa pa. What if you have 2n equals 6? So ano itsura niya? Is it going to look like, ito, magdodrawing ako guys for example. Is it going to look like this? Or, so say for example, okay, is it going to look like this? Or is it going to look like this? So this is the first chromosome. This is the second chromosome colored in purple. All right, okay. Second chromosome, sorry. And this is the third chromosome. Here. Okay, so which between the two images you see here, so this is image 1 and this is image 2, reflects 2n equals 6. Sige, tingin muna tayo sa first na aspect, 6. So ilang centromeres makikita natin? So 1, 2, 3, 4, 5, 6. So 6 centromeres. So far, makikita natin na 6 centromeres siya, yes. And at the same time, eto. 1, 2, 3, 4, 5, 6. So eto, 6 centromeres, 6 chromosomes din. Okay. So tingnan natin at the same time, yung left side of the equation. So how many sets do you see? So this is one set. This is another set. Hala, may pangatlong set siya. So ano siya bale? So this reflects 3n equals 6, which is not good. Whereas if we compare this with this diagram over here, so we have one paternal set and one maternal set as shown here. So this is more likely the case of 2n equals 6. So given 2n equals 6, anong tawag natin sa bawat isa nito? These are referred to as your homologous pairs. Or also known as your bivalents. Okay? So your homologous pairs or your bivalents simply reflect the number of chromosomes that exist as pairs. Okay, so this is one pair, another pair, and another pair. Since you have 6 chromosomes with 2 sources, so 6 divided by 2 gives you 3 homologous pairs or bivalents. I hope we're clear. So far, so good, guys. Klaro ba tayo? So far, so good. Pa-reacts gaming naman, mga friends. If medyo na-gets yung konsepto, pa-thumbs up. Ayan, pa-thumbs up. Pero kapag medyo blurry pa yung concept, pa-iyak emoji. Kamusta? Okay, I'll take that as okay. Anyway, so let's proceed with the concept of, by the way, let's review. So your chromosomes and your chromosome number. So I hope we're clear. Okay, let's take a look at the human karyotype this time. Yung human karyotype nyo, so since our chromosome number is 2n equals 46. So 2n equals 46 means that kung divide natin to, 46 divided by 2 equals, you have 23 homologous pairs. So the 23 homologous pairs is composed of these chromosomes labeled in pink, as shown here. And the last pair, so eto sila, 22 yan siya. And the last pair happens to be very special. Because the last pair can either be an XX or an XY chromosome. And this is referred to as your sex chromosomes. And these are very important in determining the secondary sexual characteristics, and at the same time, the sex of the individual. So this is the human karyotype. As we can see, 46 chromosomes. Kung bibilangin natin yung number ng centromeres, makikita nyo, 46 yun siya lahat-lahat. There, 46 chromosomes. So on and so forth. All right? So this is the human karyotype. When you say karyotype, this is simply referring to the spread of all the spread wherein you see the chromosomes all arranged from chromosome 1, which is generally the longest, all the way to the last pair, that is your sex chromosome. Yan siya. So that is what you refer to as your karyotype. So I have this question. Which do you think in these 4 images reflect a chromosome? Alin dito yung chromosome? Is image A a chromosome? B a chromosome? C and D chromosomes? If your answer is C, kasi nakita nga kanina, na-discuss nga kanina, then you are correct. B, and D are also chromosomes existing at different times in the cell cycle. So what does this tell us? Your chromosomes are structurally dynamic. Hindi ibig sabihin na all the time, ganyan lang yung itsura niya. Your chromosomes are structurally dynamic as it progresses through the cell cycle. So titingnan natin therefore yung cell cycle. Sir, ano yung cell cycle? Your cell cycle simply reflects, wait lang, mukhang lumiit yung screen ko. Ayan. The cell cycle simply reflects what happens during the life cycle of a cell. From its birth after mitosis, all the way it has committed, all the way up to the point that it has committed, to cell division at the M phase. Ayan siya. Oops, sorry. Yung ano ko. Sorry. There you go. Okay, so if we are going to take a look at this diagram, this simply reflects kung ano nangyayari sa cell. So say for example, at these specific points, you can specifically divide the cell cycle into 2. So the first half here, or the larger chunk that is being traced by my highlighter over here, is referred to as the interface. Kapag sinasabing interface, this is the non-dividing part of the cell cycle. Cell cycle, CC na lang, of the cell cycle. Whereas the M phase is the point in the cell cycle, M phase, is the point in the cell cycle that it has committed to cell division. So at this point, kapag nag-step in na siya sa M phase, automatically it will commit to cell division. Hindi na siya aatras. Ika nga, in the words of our broken-hearted friend, sana all committed. So M phase is a committed step. Ibig sabihin, hindi na siya pwedeng maibabalik pa. Ala, okay. So titingnan natin ngayon kung ano yung nangyayari sa chromosomes between the interphase and the M phase. So for the sake of discussion, we will take a look at an example wherein yung chromosome number is 2n equals 4. Okay? So 2n equals 4. So therefore, yung itsura ng chromosome natin, so you have these 2 first and these 2. So ito yung example natin for this particular discussion. So 2n equals 4. So 4 chromosomes. Tama, there are 4 centromeres, 2 sources diploid. Tama. So we will start with the discussion. So at G1 is the first step in the interface. And at G1, yung commitment lang ng cell is with cell growth and in performing its metabolic function. So kapag sinasabing metabolic function, it means that it will simply perform what it is intended to perform. Say for example, if it is a goblet cell, then it will produce mucus. If it is a muscle cell, then it will simply contract. So that is G1. So at G1, your chromosomes will look like very thin threads, like this. Thin threads pa yan siya. Later, we will see bakit dyan siya nagsimula. Ayan siya. So at this point, your chromosomes will exist as very thin threads we refer to as chromatin. Ayan siya. Para hindi kayo mawala, chromosomes na manipis, chromathin, chromatin. Okay, so at this point, your chromosomes will look like this. Bilangin natin number of centromeres. 1, 2, 3, and 4. So therefore, chromosome number 4, left side of the equation, how many resources? 1 and 2. So therefore, 2n equals 4 pa rin siya, regardless of the appearance your chromosome takes. So 2n equals 4. Here, after it has gathered enough resources for it to go through the next step, pwede na siya magko-commit to proceeding with the S phase. Sa S phase naman, this is referred to as the synthesis phase. Because at this point in time, a very important process occurs. This is where DNA replication happens. So with DNA replication, kung ano yung instruction na makikita natin sa isang chromosome, kasi remember guys, etong isang strand ay tad-tad ng information we refer to as gene loci. Kokopyahin niya lang yan. Kokopyahin niya lang yan by virtue of DNA replication. So bali kinopya niya lang guys yung information on both strands. So we can see the difference from how it appeared during G1 to how it is now during G2. So in short, parang dumoble yung ating genetic content. So if we will take a look at this example, we can see evidently that there is DNA replication because of the presence of a new structure. This new structure is referred to as a sister chromatid. Ito siya. So if this highlighted region is a sister chromatid of this highlighted region. In short, these two are carbon copies of one another. Okay, so you now see sister chromatids at this phase, but let's try to count the number of chromosomes. So the number of chromosomes again are counted according to the number of centromeres. 1, 2, 3, and 4. So at this point, chromosome number is 2n equals 4 pa rin. So this is one importance. This is one important aspect of the interface. You double the DNA content as shown in the prevalence of your sister chromatids, but the chromosome numbers will always remain the same. Okay? So we double the DNA content, but the chromosome number remains to be the same. Okay. So after DNA synthesis and replication, it will simply go back to its metabolic state and then to its hypermetabolic state, and then it will prepare for mitosis at G2. So at G2, makikita natin that your chromosomes will now start to condense very lightly. Ayan siya. So magko-condense na siya ng very light. Ayan. So the same is true with the second homologous pair or the second bivalent. So this actually represents the chromosomes starting to prepare itself for cell division. So here we can see the chromosome number to still be at 1, 2, 3, 4. 2n equals 4. Equals 4. And then here we can see the synthesis of proteins necessary for cell divisions. For cell division. There you go. So synthesis of proteins necessary for cell division. So makikita natin yung journey ng chromosomes natin. So pero parang medyo nakukulayan na itong mga images na to, no? So here, and then once it has fully committed itself to mitosis or meiosis, then we can proceed with cell division na. But so far so good guys. Klaro ba tayo sa concept ng interface? Interface muna tayo. So yung may tatlong very important aspects dito guys. Number 1, G1, metabolic function and cell growth yung priority, and your chromosomes will exist as chromatin. S, it means that the cell already has gathered enough resources for it to double its chromosome, to double its genetic content thereby producing, ano ulit tawag dito? Sister chromatids, which are actually carbon copies of a single chromosome. Also, makikita din natin that the chromosome number still remains to be the same. After it has completed S phase, we proceed with G2, and in G2 the cell will still be metabolically functional, but at the same time it will also start to revert its focus to preparing itself for cell division. So here makikita natin yung chromatin threads natin magsa-start na yan siya mag-condense. And then afterwards we proceed with mitosis or meiosis. So in M phase, sulat ko muna dito guys. Your M phase can be specifically identified as mitosis or meiosis. Mitosis occurs in body cells, whereas meiosis occurs in the production of sex cells. So later, or as we go through the process of discussing mitosis and meiosis, makikita natin yung very important differences. But for now, we start with mitosis. So assuming that the cell we discussed a while ago is not a cell responsible for the production of gametes, so it will undergo mitosis. Okay, so mitosis is divided into 4 specific phases. So these are your prophase, prophase, metaphase, anaphase, and lastly, your telophase. Okay, ano nangyayari? So, yung entire point ng process ng mitosis is that it has to undergo all these four stages so that it can produce 2 identical daughter cells. Okay, so we start with prophase. Remember, our chromosome number a while ago is 2n equals 4, right? 2n equals 4. So at this point in the cell cycle, makikita natin that your chromosomes are already fully condensed. So fully condensed na yung chromosomes natin, as shown here. There. So makikita natin that 2n equals 4 pa rin siya because if we count the number of centromeres, 1, 2, 3, 4, all right? And also, one very important physical manifestation of prophase is the fact that at this point in time, you will you will notice that the nuclear membrane will start to disintegrate. So remember guys that your chromosomes are confined in the nucleus. And since the process of cell division will generally distribute the chromosomes into 2 daughter cells, it has to break the compartment that contains it, and that is your nucleus. So nuclear membrane will start to disintegrate, and your centrioles will now start to form. So yung centrioles mo guys are very important because these are the ones for chromosomal migration. Chromosomal movement na lang guys, to be sure. For chromosomal movement. So these are your centrioles. So sila yung nagpo-produce ng microtubules necessary to move the chromosomes around. Okay, so yan yung nangyayari at prophase. The chromosomes are now fully condensed. The nuclear membrane is now starting to fully disintegrate. And at the same time, you observe the presence of centrioles starting to take shape because these are the ones necessary to organize the microtubules necessary for chromosome movement for the later stages. There, so that's what happens at prophase. At metaphase naman, your chromosomes will now start to individually align at the metaphase plate. So notice how alignment is individual in nature. So paisa-isa silang pipila sa metaphase plate. So the metaphase plate is not actually a structure, but instead this is just a hypothetical region kung saan nag-a-align yung chromosomes natin. Alright, so alignment happens this way. and the chromosomes will align. Take note of the term. Very important to siya, guys. This is one of the major differences between mitosis and meiosis. Metaphase, during mitosis, alignment is individual. Later, makikita natin sa meiosis kung ano nangyayari. Kung bakit siya different. And also, the centrioles that we saw a while ago colored in pink have already fully migrated towards the opposite poles that it has established. And at the same time, the spindle fibers, which are microtubules in nature, will be associated and will attach themselves to the centromeres. Kaya nga guys, sinabi ko kanina sa earlier discussions natin that the centromeres are necessary for chromosomal movement. Dito na siya makikita kung bakit. Because this is where the spindle fibers will attach. Okay, so that is what happens at metaphase. Okay, so bilang tayo ng chromosomes ulit. At this point, 2n equals 4 pa rin siya. Let's verify. How many centromeres do we find? 1, 2, 3, and 4. 2n equals 4. Okay, so here, as it proceeds with metaphase, or rather, as it finishes metaphase, the next step is for it to proceed to anaphase. Eto guys, whenever you are dealt with a problem with cell division, the only way for you guys not to get lost is to appreciate the fact that whatever you align at metaphase, you will separate at anaphase. So since you have aligned individual chromosomes here, therefore, you will separate these individual chromosomes according to the sister chromatids at anaphase. So kung titingnan natin to, so the first chromosome here is red. So bale, ayan siya. Magsa-separate na yan siya bale. So there is now the actual separation and movement of your chromosomes. Oh, sorry. Ayan siya, guys. Here we see that your chromosomes are now fully separated according to its sister chromatids. And this is made possible because of the spindle fibers that pull them apart. So, since you aligned individual chromosomes at metaphase, you have separated these individual chromosomes at anaphase according to its sister chromatids. So now the sister chromatids na nakikita natin kanina are no longer existent because these are now functionally termed as daughter chromosomes. So at this point in time, bilangin natin yung number ng ating chromosome. So eto, dito na siya medyo magkakatalo. So in anaphase, because you now have established a pole. So here, say for example, this is pole A and this is pole B. So let's count the number of chromosomes. Specifically, daughter chromosomes. So 1, 2, 3, and 4 for pole A. So chromosome number is 4. How many sets? Maternal and paternal. 2n equals 4 at this point in time. At anaphase during sa pole B, 1, 2, 3, and 4. So chromosome number 4. And then how many sets? You have a maternal set and a paternal set. So 2n equals 4 pa rin siya. Okay? So makikita talaga natin dito guys that this is referred to as equational division. Bakit siya tinatawag na equational division? Equational division siya in the sense that we started with 2n equals 4 kanina and the resulting products will still receive the same chromosome number 2n equals 4 happening during mitosis. This is also one very critical difference between mitosis and meiosis. So at telophase, actually, yung nangyayari lang during telophase is the formation of the new daughter cells. So yung makikita natin dito is that after the end of the process, you will now start to see the formation of the new nucleus at the ends of the dividing cell, and at the same time, your chromosomes will now start to relax. So magre-relax na yung mga chromosomes natin. I-duplicate na lang natin to guys para hindi na tayo mag-drawing ulit. Yan. There. So your chromosomes will now start to relax at telophase. So at telophase, if we will count the number of chromosomes, it will still be 2n, it will still be 2n equals 4 per daughter cell that you produce. Per daughter cell that you produce. So here makikita na natin the process of full movement of your chromosomes towards the opposite poles, wherein it establishes the new nucleus of the dividing cell, and at the same time, hindi lang yung ating chromosomes yung mag-move. Your cellular organelles will also move and redistribute themselves to the new daughter cells being formed. So to summarize this, at the end of mitosis, you will produce 2 daughter cells with identical chromosome numbers. Ayan siya. So guys, take note to kung ano yung itsura ng chromosomes natin at this point. Doesn't this look very familiar to you? Diyan din siya nagsimula at G1. Because after telophase, the process will now begin anew with G1, S, and G2. And then once it has proceeded with M phase, proceed ulit siya sa mitosis. So this is very cyclical, hence cell cycle. All right, so before proceeding to meiosis, kakamustahin ko muna kayo. Guys, kamusta? Kamusta? So far, so good? Pulso ng masa. If okay pa guys, pa-thumbs up or pa-heart or kahit ano. Or if may questions man, now's the time to raise them. So far, so good? All right, sige, I'll take that. Okay. So let's proceed with the next type of cell division. The next type of cell division is very similar to mitosis with very striking differences. So similar but different at the same time. So makikita natin to. So at meiosis, the main objective of meiosis is to produce gametes. Okay, so remember that your gametes or your sex cells are haploid in nature. And meiosis itself will require 2 rounds of cell division, such that we start with a single daughter cell, a single parent cell here. After 1 round, you produce 2, and after the next round, you produce 4. So this is one of the major differences with mitosis. Kasi sa mitosis pa lang, 2 daughter cells lang yung mapoproduce. Whereas sa meiosis, you produce 4 daughter cells, and at the same time, from N na diploid state, magiging haploid yan siya, guys. So very important aspect ng meiosis, yung production ng haploid cells. As compared to mitosis, that the diploid parent cell will produce diploid daughter cells as well. So dito makikita natin may haploid din na nagaganap. So etong pansin nyo, madaming circle sa meiosis, no? So ibig sabihin yan, marami tayong mapag-uusapan. So yung meiosis, remember, 2 rounds of cell division yan siya. So ibig sabihin, it will undergo prophase, metaphase, anaphase, and telophase twice. That's the main reason why malalaman mo kung meiosis or mitosis yung tinatanong, kasi may Roman numerals yan siya at the start to indicate at which cycle na siya ng meiosis. Is it in the first round of meiosis or in the second round of meiosis, as indicated by Roman numerals 1 and Roman numerals 2. Just like mitosis, may prophase, may metaphase, may anaphase, and telophase din siya. So here, let's start with prophase. So since specific na tayo sa meiosis, prophase 1 siya dapat. Okay, sa prophase 1 guys, there are very important events happening. So in prophase 1, this is further divided into leptotin, L, Z, P, D, D, which stands for leptotin, zygotin, or in older books, zygonyma, pakitin, or in older books, pakinima, diplotin, or in older books, diplonema, and lastly, diakinesis. Or in older books, diakinesis. Nothing special. Okay, so we start with leptotin. So sa leptotin, let's discuss prophase 1 from a perspective of a single homologous pair or a bivalent. So, pagod tayo. So sa leptotin, what happens here is that your chromosomes will exist as thin strands. Kaya lepto, ibig sabihin yan from its derivation or its etymology, it simply means thin strands. So your chromosomes will exist as relatively thin strands pa. So this is one bivalent. Yan siya. Alright, so thin strands. And then at zygotin, one very important event happens. This is where synapsis happens. Synapsis. This is where synapsis happens. Kapag sinasabing synapsis, homologous chromosomes will start to, duplicate na lang natin to guys, will start to approach each other on a gene-to-gene basis. So, ang ibig sabihin lang yan guys, is that at this point, so yung blue natin dito, will pair and synapse with its homologous pair. Ayan siya. So magkakalapit lang sila at zygotin. At pakitin, you will observe that the chromosome, that the arms of the chromosomes, arms of the chromosomes will now start to overlap. So mag-overlap na yung arms of the chromosomes natin. So, say for example, ito, pwede siya mag-overlap with this and this. So this overlapping part here will actually be the site where crossing over will occur, which is completed at diplotin. So at diplotin, kung ano yung site wherein the chromosomal arms overlap, this is where crossing over is finalized. So when you say crossing over, this simply refers to the process by which there is chromosomal exchange in the segment. So yan siya. Mag-exchange lang yung segment such that at the end of the process, you will produce two chromosomes that are now recombinants of each other. Alright, so makikita na natin na may mga differences na, no? So this is actually very important as this provides more avenue for variation. So ayan siya. So this is where crossing over occurs. So yan yung important talaga sa prophase 1. It simply tells you that crossing over will occur in homologous pairs or bivalents. Alright? Ay, may nagtanong ata. May tanong ko po, ano po na app yung nagdo-drawing ka po, sir? Sabi ni Ma'am Grace. This is Notability and this is available sa iPad. Free lang siya actually. Okay, so tapos na tayo sa prophase. We now start with metaphase. Sabog na daw yung utak ng isa nating viewer. Okay lang yan guys. Magkakaroon naman tayo ng break later. So at metaphase 1, remember, anong ulit yung nangyayari sa metaphase? This is where chromosomes will align at the center or at the metaphase plate of your dividing cell. So remember that we are looking at the entire experiment from 2n = 4 perspective. So 2n = 4. The same din siya with our mitosis kanina. 2n = 4. So at metaphase 1, your chromosomes will align at the metaphase plate. That's true. But yung very special na nangyayari dito is that, so since may mga crossing over events na tayo dito, guys, instead of aligning individually, your chromosomes will align as homologous pairs, as shown here. So, pwede na magkabaliktad yan siya. Ito. So, for example, in our experiment here, walang crossing over na nangyari sa second homologous pair natin. So, mapapansin natin na at metaphase 1, one major difference here is that instead of the individual chromosomes aligning at metaphase 1, what you align at metaphase 1 are actually bivalents. Because remember, these are bivalents. So you line up homologous pairs or bivalents at the metaphase plate. So ito siya, metaphase plate natin to siya. There. And also the same characteristics yung makikita natin. You still have the centrioles here being that arranges the spindle fibers. So yeah, so metaphase 1. So if we're going to count the chromosomes, 1, 2, 3, 4, still 2n equals 4. Okay. So, we then proceed with anaphase 1. So, at anaphase 1, remember ano nga yung rule natin sa anaphase and sa metaphase? Whatever it is you align at the metaphase plate, you will separate at anaphase. Kung ano yung pinagtagpo sa metaphase, yun yung ipaghihiwalayin sa anaphase. So, are you guys at metaphase or are you guys at anaphase? Char lang. Anyway, so titingnan natin to. So, whatever you have aligned, you will separate. So, if we will take a look at this, so isa-separate na natin yung bivalents. Yan siya. So, dapat medyo makatotohanan kasi parang kinakaladkad na yung chromosomes natin. Ayan. There you go. And then your other bivalent. I-draw din natin siya. Okay. And at the same time, your spindle fibers, very important. And here. So this is what happens at anaphase 1. So at anaphase 1, let's try to determine the number of chromosomes per pole. So pole A and pole B. So let's count the centromeres. 1 and 2. Hala, dalawa na lang. Sige, pagpatuloy natin. And at the same time, let's count sets. I only have one set for this bivalent and another set for this bivalent. So, n equals to 2 na lang siya. At the same time, the same is true with the other pole. Alagay mo ka na. Ayan, okay. That's right, that's right. N equals to 2. So, dito, makikita na natin yung sinasabi natin na this is referred to as, sorry about that, wait lang, reductional division. Ayan, reductional division na siya at this point because if we compare it to equational division during mitosis, 2n equals 4 will produce 2n equals 4 and 2n equals 4. But in anaphase 1 of meiosis, 2n equals 4 has produced n equals 2 and n equals 2 na cells with their respective chromosome number. Dito natin makikita yung reduction ng chromosome number in the formation of your gametes. This is the main reason why all of the cells in your body are diploid except for your gametes. Because your gametes undergo meiosis, and meiosis enables 2 rounds of cell division, and the first round of cell division will have the chromosome number. So say for example, if tao to, so we start with 2n equals 46 for the parent cell. If this is meiosis, the first round of cell division will produce only n equals to 23 per daughter cell, which will proceed with the next process. So we proceed with anaphase, and eventually this will proceed to telophase. So dito guys, telophase 1. So dito nagkaklaro na talaga kung ano yung behavior ng chromosomes during cell division and during G1. So let's take a look at this. So sineperate na nga natin yung chromosomes during anaphase 1, di ba? So eventually, this will just further migrate towards the end of the cells as shown here. So mag-full migration lang yan siya guys and nothing else na. Kasi in meiosis, very transient and very quick lang yung telophase tsaka yung prophase. So remember, may recombinant part tayo dito. Over here. There. Yeah. So makikita na natin na ito na yung nangyayari sa chromosomes natin at this point. So telophase, and eventually after telophase, let's count again the chromosomes. So this will produce n equals to 2 and then n equals to 2 in this particular cell over here. So we proceed with prophase 1. At prophase 1, walang masyadong critical na nangyayari, guys. At prophase 1, the cell will just prepare itself for another round of cell division. Sorry, prophase 2 na siya balik kasi second round of cell division na siya. Okay, guys, let's review muna ano yung nangyari sa first round ng meiosis. Meiosis, first, prophase 1. Prophase 1, pinaka-importante na nangyayari dito is hair crossover events, which will produce variation, genetic variation. And after prophase 1, wherein crossing over is occurring, we proceed with metaphase 1. At metaphase 1, remember, important yung chromosomal alignment. But in meiosis, what aligns here are the chromosomes coming together as bivalents. So your homologous pairs will align at the metaphase plate. And whatever you align at the metaphase plate, which is your bivalent, will be separated in anaphase 1. And in anaphase 1, as it separates, you produce a reductional division wherein you have the chromosome number, proceeding with telophase 2. And then after telophase 2, we will proceed with telophase 1, with prophase 2, sorry. And then at this point, the cell will simply prepare itself for another round of cell division, as shown here. Ayan siya. So at this point, so kung kung prophase 2 na to, dapat fully divided na yung cell natin. Yan, fully divided na siya. So let's try to count the chromosome number 1 and 2 per cell. Sorry. Ito. N equals to 2 per cell. And n equals to 2 for this particular cell. So we start with prophase 2 this time. So at prophase 2, we can then proceed with metaphase 2. Ano nangyayari sa metaphase 2? This time, instead of aligning chromosomes differently, you will align the chromosomes individually, just like mitosis. So yung chromosomes natin kanina will align like this. So your other homologous pair will align like this as well. So this time, notice how alignment is individual, and at the same time, yung ating spindle fibers will be arranged by your centrioles. Okay, so at this point, let's count the chromosome number. So this will be this will be n equals to 2 and n equals to 2. Let's verify kung n equals to 2. One centromere, another centromere, just 2 chromosomes, hence this part here. And how many sets? Only one set for the first homologous pair and only another set for the second homologous pair. Okay. So therefore, just one n, or simply put, n. Okay, so at metaphase 2, we proceed with anaphase 2. So what happens at anaphase 2 is, remember, whatever you aligned at metaphase, you will separate at anaphase. So we can now separate your chromosomes individually, as shown here. Ayan, may mga recombination events tayo na dapat makita. Here, and then you will also still observe your spindle fibers that will drag your chromosomes towards the opposite poles. Ayan siya. So, per pole per cell, kung titingnan natin, so this is n equals to 2, n equals to 2, n equals to 2, and n equals to 2. And as it proceeds with the process of telophase, ma-full separate mo lang yung cells, thereby producing 4 different cells here. So this is one cell with n equals to 2. This is another cell with n equals to 2. Another cell with n equals to 2 and n equals to 2. So bale, mabivisualize na natin that we started with 2n equals 4. And after the first round, n equals to 2. So dito natin makikita yung reductional division. Specifically during anaphase 1. And here, n equals to 2. Na cell division, which gives us 4 daughter cells with a haploid chromosome number as compared to its starting parent. Okay? So this is what happens during meiosis. So ayun, let's compare and contrast. So sa mitosis, kung anong inalign mo at metaphase, makikita natin individual chromosomes. What about meiosis? At metaphase 1, yung inalign natin are bivalents. What else? Mitosis, major difference. At anaphase, what you divide are your chromosomes on the basis of their sister chromatids. At meiosis, during anaphase, you separate the bivalents. You separate the homologous pairs. All right. And also here you will observe reductional division. Let's take a look at photos, the fun part. So here is an onion root tip slide. So kung onion root tip, is this picture undergoing mitosis or meiosis? What do you think? Chat box. Mitosis or meiosis? Mitosis siya in the sense na bakit siya mitosis? Because this is not producing gametes. This is very important in simply elongating the root tip of the cell. So here makita natin mitosis. What about this one? This is a very interesting picture I came across last night while reading journals. Tapos nakikita natin dito, very weird. Ito nga sabi, atypical mitotic figures. Tapos nakikita natin na very weird yung behavior ng chromosomes natin. So this often occurs a lot, specifically in multinucleated cells, such as cells of the immune system. And at the same time, this is also very prevalent in cancer cells. Here, what do you think? See, analyze the image. Tell me if it is mitosis or meiosis. Is this mitosis or meiosis? Yung makakasagot, bibigyan ni Sir Josh ng bahay at lupa. Biro lang. Meiosis, sabi ni Ma'am Desena. Si Jay also agrees. Anna also agrees. Good job. Paano niyo nasabi? Anybody? Guys? Reductional? Actually, medyo mahirap sabihin na reductional kasi hindi natin mabibilang yung chromosomes at this point. Pero tama ka na reductional division to. Yung pinaka mag-give it away is the fact na this is the first round of cell division and also this is the second round of cell division producing 4 daughter cells. No, yun yung pinaka mag-give away talaga sa kanya kung meiosis siya or mitosis. But thank you, Anna, for saying that this is reductional because you are correct. Okay, So let's try to entertain questions prior to proceeding. Hi sir, ask ko lang po, ano po ibig sabihin ng sinasabing recombination? Ano nangyayari doon? Thank you po, sabi ni Jean. Okay, so kapag sinasabing recombination, this is simply crossing over. Di ba, at prophase, specifically prophase 1, crossing over occurs. So ano nangyayari sa crossing over? So say for example, I have here a member of a bivalent. So here. So at crossing over, there is chromosomal exchange between these two, such that you will produce recombinant chromosomes at the end of the process. Ayan siya. Wherein instead of just containing one type of strand in the process, you will produce chromosomes that will be products of recombination. So makikita natin dito na there are segments coming from another arm of another chromosome, specifically that of its homologous pair. As during anaphase, what caused the pulling of spindle fibers to separate the chromosomes? Okay, so the spindle fibers are generally directed by your centrioles. And these centrioles play this function in organizing the spindle fibers. And on top of that, your microtubules have this contractile ability because they are also proteins in nature. So they have the ability to expand. They also have the ability to contract, just like any other cytoskeletal component in the cell. So kaya ayun, may capacity siya to expand and contract, to pull the chromosomes towards its direction. And that direction is centralized and gravitated towards your centrioles. All right? Very interesting questions and very smart questions at the same time. Thank you for participating. Okay, so are we clear so far, guys, with mitosis or meiosis? Clear siya? Okay, so let's proceed with Sir Josh. Magbe-break ba muna or proceed tayo sa next topic? Mendelian principles. Kaya niyo pa ba, guys? Medyo mabigat-bigat to yung
Kayo pa, sir. Pwede naman pa siguro.
Sige, kahit isang Mendelian principle lang prior to taking notes. Okay. So, nakita na natin kung paano nagbe-behave yung chromosomes natin during mitosis and meiosis. And more importantly, mas na-discuss natin what happens during meiosis kasi yung process of producing the gametes is very central to what happens in the next generation. Kasi doon natin makikita kung ano yung nangyayari sa chromosomes na ipapamana natin sa next generation. So Mendelian principles actually rely on the concepts discussed in the previous topic. So dito makikita natin kung paano niya naapektuhan yung heredity. So prior to proceeding, let's first define a lot of terms that will be used from time to time as we discuss the law of segregation and the law of independent assortment as Mendelian principles. So first, when you say a trait, this simply refers to a general term that will describe a physical characteristic. All right? Or a physical character. Say, for example, flower color. Flower color in pea plants. Okay, so this is a trait. So these traits are generally coded by genes. So the genes can exist as two versions, which we refer to as your alleles. So when you say alleles, these are alternate versions of a gene. So say, for example, flower color in pea plants are dictated by your genes, and there are different alleles that control it. Say, for example, one allele is said to be dominant, while another allele, say for example, hindi pala yellow yung flower color sa plant. Purple pala siya versus white, sorry. So say for example, a gene which we will indicate as P will code for purple, and its allele or an alternate version of the gene that codes for flower color will code for white. So both are coding for flower color, but these are alternate versions of each other. Merong isang purple, merong isang white, but they still code for the same trait. So these are referred to as alleles. So this is referred to in the case of Gregor Mendel as the dominant allele. And this is referred to as the recessive allele because your alleles will generally come in pairs. So given these two types of alleles, there are multiple pairs that can be produced. One pair that contains both the purple dominant allele, which we refer to as the homozygous dominant. Another pair, which will comprise one purple allele and one white allele, which we will refer to as a heterozygote or heterozygous in nature, and one pair that will contain both alleles for the recessive. So this is homozygous recessive. All right. So these are referred to as your genotypes. So these genotypes represent the set of genes or the set of alleles that you have for a trait. So there. So that this is your genotype. And actually, your genotypes will code for the physical manifestation of the genotype, which is referred to as the phenotype. So the phenotype for an individual containing this genotype is purple. And at the same time, if you're a heterozygote for the trait, you will also contain the purple flower color here. Whereas if you contain the allele for, or rather, if you contain the homozygous recessive genotype, then you will produce white flowers. The physical manifestation of your genotype is referred to as your phenotype. Okay? So clear na sa atin yung konsepto ng alleles, yung genotypes which occur as a homozygous dominant, heterozygous, and homozygous recessive, and the concept of phenotype, that is the physical manifestation of a genotype. Okay, so these terms will be used from time to time as we go through the Mendelian principles of heredity. So, the first Mendelian law is the law on segregation. Remember that your genes will occur in pairs. So you have one copy of the gene coming from your mother and another copy coming from your father. So say, for example, a pea plant here. So say, since you have a gene that occurs in pairs, so ano yung pwedeng mangyari dito? The law of independent segregation tells you that the genes or that occur in pairs will be separated during meiosis. Ano lang ibig sabihin nito, guys? Say, for example, I have here an individual that is a heterozygote. So I have a pea plant here that produces purple flowers, and I have distinguished that plant to be a heterozygote. So the law of independent segregation tells me that the gametes this will produce, say for example, this is a male, therefore the sperm cells that this will produce will carry either the dominant P na allele or the recessive P na allele, because the gene pairs will be separated cleanly during meiosis. Are we clear? So, paano to siya nagma-manifest during meiosis? So, say for example, I have here a chromosome and this chromosome will hold the information for flower color. So one is coming from my father and another is sourced from my mother. So, ayun siya. So, say for example, the chromosome coming from my mother codes for the dominant P na allele. And the one coming from my father codes for the recessive P na allele, which explains why you have a heterozygote here. Okay? So, kung titignan, yung kanyang genotype is simply this. Sir, bakit twice nag-appear yung big letter P and twice nag-appear yung small letter P? Because remember, during S phase of the mitosis, idodoble mo lang yung DNA content mo. So, yan siya. Okay. So, here, makikita natin that you have an individual that is a heterozygote. Paano to siya magli-lead into producing a sperm cell that is only carrying a dominant P or a recessive P? So, as it undergoes meiosis, ano nangyayari? So, at meiosis, draw tayo ng circle dito. So, at meiosis, your chromosomes, remember ano ulit yung nangyayari sa chromosomes natin? Your chromosomes will align at the metaphase plate as shown here. So, duplicate din natin ito isa. Oy, sorry, ayaw na niya ma-duplicate, guys. Here. So, your chromosomes will align at the metaphase plate here, as you can see. And then, eventually, these will separate cleanly during meiosis. So, ayan siya. This will produce this cell at the end of the process. So, here, ito yung maproproduce mo na isa. And this one, ito yung maproproduce mo na isa. Alright, and after one round of cell division, magde-divide ulit yan siya, thereby producing 4 daughter cells, 1, 2, 3, 4, that will carry either the dominant P or the recessive P. That is the main reason why this particular individual will produce only 2 types of gametes. One that carries a dominant P and one that carries a recessive P. Very interestingly, nung time ni Gregor Mendel na naisip niya to, wala pang microscopic basis to prove his point. All he used was statistical measures. So yan siya. So yan lang talaga yung pinaka-bottom line ng segregation, guys. Segregation tells you that the genes that occur in pairs will be separated cleanly during meiosis, such that each gamete that you produce after meiosis will only receive 1 copy of the gene pair. Okay, sample tayo. Say for example, I have an individual that is homozygous dominant for the trait. So what type of gametes will this individual produce? The gametes that he will produce will carry what type of allele? Ano yung i-carry niya? Chat box? This will only carry the dominant allele. So say for example, this is a female, so all the egg cells will carry the dominant allele only. Okay? Kasi kung isa-separate mo yan siya cleanly, and then during meiosis, thereby making sure that each gamete produced will be carrying just 1 type of allele. So, alam naman natin na kung heterozygote yung individual, the gametes it produces will either carry the dominant P or the recessive P with equal ratios. What about, I have here a homozygous recessive. Ano yung mga gametes na mafo-form? This will only produce gametes that will carry the recessive allele 100% of the time. All right. So clear tayo guys sa konsepto ng ng segregation. So this is segregation. Your genes will occur in pairs. Yes. But as you produce your gametes, this will be halved such that you will only carry 1 allele per gamete. Okay? What about independent assortment? So independent assortment naman, kung kanina tinignan natin what happens within the gene pair. This time, let's take a look at among multiple gene pairs. So, by definition, state muna natin yung definition ng independent assortment. Yung definition ng independent assortment means that alleles from different gene pairs will assort independently from one another. So say for example, classic na makikita natin dito is a case of a double heterozygote. Because remember, different gene pairs na siya. So this is best visualized by looking at multiple gene pairs this time. Say for example, you have here a heterozygote for the gene pair A and a heterozygote for gene pair B. Kung titingnan natin by definition, an allele from different gene pairs, say for example, the allele from dominant A, from the gene pair A, will assort independently with any allele of the second gene pair. So may equal chances siya of assorting with a dominant B and equal chances of assorting with a recessive B. At the same time, the recessive allele from the first gene pair can also assort independently with a dominant B and a recessive B, such that the possible gametes that you will form from these individuals will be the following. So you have a dominant A assorting with dominant B, a dominant A assorting with recessive B, a recessive A assorting with dominant B, and a recessive A assorting with recessive B. So here are the possible gametes that you can produce out of this individual in terms of two gene pairs. Okay, so tingnan natin. Ano yung kaniyang microscopic basis or ano yung kaniyang cytogenetic basis? So say for example, the first gene pair here, ito siya guys. So ito yung magiging basis natin. Ito. One sec. Okay, so say for example, this chromosome here carries with this dominant A. This chromosome carries with it dominant B. This chromosome carries recessive A. This chromosome carries recessive B, thereby explaining this genotype here. All right? So let's take a look at what happens during meiosis. So during meiosis, so magdodraw tayo ulit dito. Here. So during meiosis, duplicate lang natin ito siya guys. Duplicate. Your chromosomes will align as such. So upon alignment, upon alignment, what we can observe is, sorry guys, hindi siya nag-cooperate. I-drawing ko na lang. So upon alignment, we can see here that, say for example, this is one possible scenario wherein the chromosome here will carry the blue and here. Okay, so this is one scenario during anong stage to guys? Anong stage? Where there is alignment. This is meiosis 1. So at meiosis 1, magse-separate such that you produce two daughter cells. So these two daughter cells carry with it the instructions for B and A, whereas this daughter cell here will carry with it instructions for B and A. So separating that further, ayan siya. So you produce this possibility that you will contain gametes that produce these possibilities. Okay, so this is one possibility only, guys. There's also this possibility that the cell during meiosis will assort this way, such that one chromosome that carries the B will assort this time with another chromosome that carries the A. I hope you can visualize this time. Ayoko na i-drawing yung chromosomes, medyo mahaba. And the dominant B here, the chromosome carrying the dominant B will assort with a chromosome carrying the recessive A. Okay? So kapag sineperate natin yan siya during meiosis, so nag-iba lang bali yung chromosome order. So yung mangyayari dito is that these two cells will produce the following. A cell that carries B and A, and a cell that carries this. And further division at the second round of meiosis will produce these 4 gametes. Okay, so this gamete here will carry the following. Sorry. Ayan siya. So makikita natin dito. Ayan. So this explains why we produce these types of gametes in the first scenario here. Medyo nabaliktad ko lang yung B and A, but they still mean the same. So here we see that the chromosome B can assort with the chromosome that carries A, and at the same time, makikita din natin that it can also assort independently with a chromosome that carries the dominant A. So in short, assortment occurs as if it's walang pakialamanan. So A here, sorry, highlighter, A here can assort with dominant B and recessive B at the same time. Recessive A can assort with dominant B and recessive B at the same time. So in short, pwede na siyang mag-mix and match during assortment. And that is independent in nature. Okay? So clear ba tayo guys sa law of independent segregation? So segregation looks at the single gene pair, and the single gene pair will segregate cleanly such that each gamete will only receive 1 copy for that gene pair. And independent assortment simply looks at multiple gene pairs this time. And the member from one gene pair can assort with any member of the second gene pair independently na walang makaka-affect sa kanya. So that is the process of independent assortment. Okay? So, guys, kamustahin ko lang kayo? So far, so good? Kamusta? Okay, I hope you guys are catching up kasi relatively medyo jam-packed yung ating topics for today. And tomorrow, relatively mas mild and mas light na siya. Sir Josh, can we take a 5-minute break kasi medyo malalim-lalim din yung next na topic?
Yes, please, Sir Eman.
Sige, sige. Ay guys, 5-minute break lang muna tayo. I'll just refill my cup of coffee and then we will proceed with the next topic. All right, kamusta? Is everybody back?
Yes, sir. Pwede po.
Okay, so since galing tayo sa very refreshing 5-minute break, do you want guys to try solving a few sample problems? Gusto niyo 'yan? Can I get an amen? Joke lang. Gusto niyo mag-try practice solving? Sige, sige. Kasi ako gusto ko. Gustuhin niyo rin. Anyway, so eto guys, so mag-share ako ng screen. So our first sample problem is relatively simple for starters. All right, so mag-share screen muna ako, friends. One sec. Your screen sharing is paused. Ayun. Okay, sample problems. So you have a dominant big letter Y indicating the dominant yellow phenotype and a recessive Y indicating the white phenotype for flower color. So this is the first trait is flower color. Ayun. And the second trait is for seed shape. Okay, so for seed shape, it can either be round, which is dominant over wrinkled, small r. Okay, so we start with a pea plant. Ito siya. With white flowers and wrinkled seed shape was crossed with a double heterozygote. So isa-isay natin. So with white flowers, ano possible? White can only be coded with a homozygous recessive. And a wrinkled seed shape. So ano yung kanyang pwedeng genotype for wrinkled? Okay, so it means that this is our first organism. So our first is YYRR was crossed with a double heterozygote. So ano yung tsura ng double heterozygote? It means that it is heterozygote for the first gene pair. So big Y, small y, and dominant R, recessive R. Okay, so yan yung cross daw nangyari. What is the probability of getting an offspring that is heterozygote? So probability yung titingnan natin or likelihood. In terms of flower color and will have wrinkled seed shape. So yung hinahanap natin dito is the probability of a heterozygote plant in terms of flower color. So ito yung first na condition. And will have a wrinkled seed shape. So ano yung tsura ng wrinkled seed shape? Ganyan siya dapat genotypically. Okay? So ito yung hinahanap natin. So with this, guys, you might be tempted to do a Punnett square. And Punnett squares aren't very effective. Say, for example, if you are given, let's say, 10 pairs of 10 gene pairs or 20 gene pairs, hindi siya super duper effective. The best way to approach this is to simply take each genotype independently. So may mnemonic kaming ginagamit dito sa UPLB. We use TEGI. TEGI. It stands for take each genotype, or rather, take each gene pair independently. Independently yan siya. Mahaba lang. Okay, so how do we approach this? So this is relatively simple. So isa-isahin mo lang yung gene pair. So the first gene pair involves the yellow color. Right. So this will be our first gene pair. Okay. So this time, let's establish the type of gametes we can produce. So for the first parent, how many types of gametes can this produce? Chat box. For the first parent, ilang possible gametes in terms of flower color? Remember, segregation, independent segregation. If your answer is all gametes will carry the recessive Y, then your answer is correct. What about for the second parent? There are two possible gametes. So the gamete can be either a dominant Y or a recessive Y. All right? So far, so good. So once you now have established the gametes, you can then proceed with the Punnett square. Okay? So the Punnett square, so ito, ilalagay lang natin yung gametes from one parent on one side of the Punnett square and the other parent from one side of the Punnett square. Okay, so the possible products of the crosses involving the first gene pair will be the following. So its genotypic ratio will be 1 heterozygote is to 1 homozygous recessive. Kasi 1 is to 1 lang siya. What about the phenotypic ratio? Phenotypic ratio is 1 yellow is to 1 white. Okay? That is for the first gene pair. Ito siya. First gene pair yan siya. Okay, on your seats, talaga. On your desks, try working on the second gene pair involving the seed shape. Okay, so let's answer together. Remember what our approach is. Our approach is to take each gene pair independently. So this and this. Tama? So what are the possible gametes for the first parent? For the first parent. Anybody? Yung sasagot, mananalo daw sa loto bukas. Alright, so the possible gametes will be R, right? And for the second parent will be either dominant R and a gamete carrying recessive R. Okay? So, may gametes na tayo na next na gagawin. We proceed with the pangit square. So, sa Punnett square natin, so we just put the gamete onto the rows and columns. All right? So, putting it in the rows and columns, here we see these possibilities. So, genotypic ratio will be 1 is to 1. All right? Phenotypic ratio this time. 1 round is to 1 wrinkled. So now let's go back to what is being asked in our problem. Okay? So what is being asked in our problem is a heterozygote and a wrinkled phenotype. So yung technique lang natin dyan is to simply highlight kung anong hinahanap. So this, ano yung likelihood na mangyayari yung isang heterozygote? This is 1/2. Tama? And also, ano pa yung second na hinahanap? Wrinkled. So this is also another half. So therefore, multiplying, so the chances of getting a heterozygote and a wrinkled out of the cross is 1/4 of the time. Okay? So very easy pa siya, guys. Very easy to sa inyo, I know. So this time, let's spice things up. Here, you have 3. And this time, you have a pair that is indicative of a sex chromosome. Remember, your sex chromosome will behave just like any other chromosome during meiosis. So you also expect them to follow the laws of segregation and independent assortment. So you have here 2 individuals, 2 humans asking for your help kung meron silang Huntington's and yung babae naman ay merong sickle cell anemia and the male is colorblind in nature, magtatanong sila sa'yo, anong probability of producing a phenotypically normal offspring? Okay? So, remember, this may appear very complicated, but just apply the rules and principles that we have used. And that is to, divine law natin dito sa genetics, take each gene pair independently and establish the phenotypic and the genotypic ratios. Okay? So let's try solving. So here, our first parent is a male with Huntington's disease but is normal for the sickle cell anemia. While our second parent is a female with normal eyesight but contains the allele for color blindness. So she is a carrier for color blindness. She is normal in terms of the Huntington's disease, yes. And at the same time, she has sickle cell anemia. They are asking, what is the probability of a phenotypically normal offspring? Knowing na medyo sakitin sila genotypically. So this is not a very good match. But let's try to check out their chances. So here, Ano nga ulit yung rule natin? Take each gene pair independently. So what's the first gene pair? The first gene pair is reflected in the sex chromosomes. So here, let's establish the crossed with, all right? Ito din ba yung ginawa niyo? Now, let's identify the possible gametes. So for the male parent, ano yung possible gametes? A sperm cell carrying the recessive N and a sperm cell carrying the Y chromosome. All right? What about for the second parent? An egg cell carrying the dominant allele and an egg cell carrying the recessive allele. Okay? It doesn't have to be super complicated, you guys, I tell you. And then, so now we have the gametes. So kapag may gametes na, ano yung next? Pangit square. So sa pangit square natin, XNY, XN, XN. Makes a lot of sense. So far, so good. All right. So, okay, so this is the first parent. This is the first possibility. The second, the third, and the fourth. Okay, so let's take a look at the genotypic ratio. So genotypic ratio is 1 is to 1 is to 1 is to 1. All right? So there are 4 possibilities in terms of the genotypic ratio. So since yung hinihingi lang naman sa atin ay yung phenotype, di sa phenotype na lang tayo mag-focus. So alin dito sa tingin nyo guys, yung phenotypically normal? So let's highlight it as green. So is the first pair phenotypically normal? The answer to that is yes. However, this individual is a female that is a carrier. Okay. What about for the second individual? Eto siya. Is this normal? The individual is phenotypically normal as well and is a male. What about for the person, for the offspring represented in the lower left box? This individual is not normal, no? So this is a colorblind female. What about for the person in the lower right? This male is also not normal. Ayan siya. So, what are the chances of getting a normal offspring for this particular cross involving the sex chromosomes? Chances, 1/2 yung normal. Clear? So 1/2 of the time. So what about for the second parent? For the second gene pair. So second gene pair, highlight natin as red. Here. So for the second gene pair, this is the cross. All right. So what are the possible gametes for the first parent? A gamete carrying the dominant and the recessive. At the same time, the female parent will only produce one type of gamete, and that gamete will carry a recessive H. Okay? So, we now have established. Okay, so eto na siya. Okay, so since the trait is autosomal dominant, it means that the normal phenotype is the recessive phenotype. All right? And the recessive phenotype will occur half of the time as well. Claro? Okay, what about sa last? Sa last naman, so let's identify the possible, ito siya, highlight muna natin. Highlight natin with dark green para hindi tayo mawala. Alright, so ito siya. So for the last pair, crossed with this, so what are the possible gametes? A gamete carrying the dominant and a gamete carrying the recessive. So lahat-lahat sila will be phenotypically normal. Alright, despite being a carrier, but phenotypically normal pa rin siya. Ay, sorry. 100%. So 1 yan siya. So what is the probability of a phenotypically normal offspring? So let's multiply the odds. 1/2 times 1/2 times 1 equals to 4. It means that with every offspring they will have, it will have a 1/4, it will have a 25% chance of being phenotypically normal in terms of being colorblind, in terms of having Huntington's disease, and in terms of having sickle cell anemia. All right? So hindi mo kailangan mag-Punnett square dito ng sandamukal, guys, kasi sobrang hirap na proseso 'yun. You just have to take each gene pair independently and produce individual Punnett squares, and that's a lot better. All right? Do I have questions coming from the audience? Questions?
Sir,
Para siya?
we have a question po from Facebook Live.
Yes.
The question po is, what if the genes of, I'm not sure if it's still included here, but what if the genes of Barr body were involved in gene segregation? What would the result be?
Oh, all right. Barr body formation. Okay, so the Barr body formation is a common occurrence in individuals carrying two X chromosomes. So generally, for gene dosage compensation, what happens is that the body will only make use of just one chromosome, just one X chromosome. So for individuals carrying the two X's or the two X chromosomes, one eventually becomes a Barr body at random. All right. So kapag ganun, gene silencing, or rather, since random yung mangyayari, say for example, can I share my screen? That is a very intelligent question. Okay. Kapag sinasabi kasing Barr body formation, what is generally observed is that, so since, kunwari, ito yung cells ng isang individual with X chromosomes. There. And physiologically speaking, the body will only make use of just one X chromosome. That is why we have this phenomena which is referred to as random X inactivation. Random X inactivation. So guys, hindi lang sa life nangyayari yung pag-inactivate sa X. Nangyayari din siya sa cells natin. Okay, so it's possible that, so kunwari, you have a gene, let's say gene 1 and gene 2 for each, kunwari, heterozygote ka for that gene. So say for example, since random X inactivation yung nangyayari dito, it is possible that for some chromosomes, ito yung ma-inactivate, while the rest, ito yung ma-inactivate. So therefore, in this case, the genes that will be expressed by the body are the genes in the chromosome that is not inactivated by virtue of random X inactivation. That's the main reason why we still observe heterogeneity across the individual. Specifically, this is commonly observed in females. Say, for example, if this is an individual, tapos this is a real life example, and sa part ng katawan na to, na-activate yung X chromosome, which is for, let's say hyperhidrosis. So sa case ng hyperhidrosis, magpapawis siya. Say for example, the person has the genes for hyperhidrosis or malalang mapagpawisan. Pero at this part of the body, yung na-inactivate na X chromosome is the chromosome that carries the gene for hyperhidrosis. So sa part ng katawan na yun, hindi siya magproduce ng so much sweat. So this is more like an exemption rather than the rule. But it's nice that somebody has brought the concept of random X inactivation concept in basic genetics. But this does happen specifically in individuals carrying an X in individuals carrying two X chromosomes. I hope I was able to answer the question, Sir Josh.
Yes, Sir Eman, thank you.
Okay naman daw po siya. Okay naman po yung nagtanong. All right. Okay. So, There you go. So that's the probability of producing a normal offspring. Okay. So, Sir Josh, until 11:30 tayo, no?
Yes, sir.
Okay, sige. Let's try to squeeze in one last topic before letting you go. And that topic involves gene interaction. So guys, just like humans, genes are not made to exist on their own. Genes are made to interact, and more often than not, these interactions produce very special effects, very special effects in the phenotype of the organism. So let's introduce the concept of gene interaction. So gene interaction, this can be determined as two. There are two types. You have allelic interaction. So this is interaction within the gene pair. And lastly, you have a non-allelic, non-allelic interaction, wherein the interaction is among multiple gene pairs. Okay, so the interaction is among multiple gene pairs. So for allelic interaction, say for example, this is common. Sige, let's start with allelic interaction first. So for allelic interaction, the interaction is commonly observed in heterozygotes that the presence of the two different alleles, two different alleles are bound to produce an effect in the phenotype. So let's discuss them one by one. So the first and the most common type is what is referred to as complete dominance. Okay, kapag sinasabing complete dominance, you have a dominant allele and a recessive allele. Just one copy of the dominant allele is enough to express the trait. Wherein in the case of homozygous dominant, it will show a dominant phenotype. In the case of a homozygous recessive, it will show a recessive phenotype. But what if it is a heterozygote? Your heterozygote will still show a dominant phenotype in the sense that one copy of the dominant allele is enough to mask the expression of the second allele. That is what we refer to as complete dominance. And generally, we indicate the dominant phenotype with a big letter or a capital letter and the recessive phenotype with a small letter. All right, so that's for the case of complete dominance. Gregor Mendel's experiments were lucky enough that his traits were simply concerning complete dominance. Because if it was the other way around, mas mako-complicate yung statistics niya and mas hindi siya makaka-arrive sa concept ng segregation and assortment. So that's for the case of complete dominance. So, say for example, I have here a parent and rather two parents, and these two parents are homozygous for contrasting traits. So what will be the offspring produced? So since this will produce a dominant A gamete and a recessive A gamete, so the offspring produced will be heterozygote. Its phenotype will resemble that of its dominant parent. All right. So here we see kung gaano ka-important yung complete dominance or gaano ka-prevalent yung complete dominance. Say for example, what if we cross this with another individual that is heterozygote as well? So the gametes produced will be the following, such that we will need a Punnett square for this. So the Punnett square will produce a 1:2:1 na genotypic ratio and a 3:1 na phenotypic ratio. All right? So this is the genotypic ratio and the phenotypic ratio. 3 indicates the probability of getting a dominant phenotype in the offspring. So, ayan siya. Okay, so that is for the case of complete dominance. Next, what about the case of incomplete dominance? Your incomplete dominance is very different as compared to complete dominance in the case that the heterozygotes are generally at the midpoint of the two extreme phenotypes of the parents. So this is commonly observed in your, say for example, yung 4 o'clock plant nyo, wherein the 4 o'clock plant, say for example, eto siya. So the 4 o'clock plant will have genes coding for red and a gene coding for white. Sorry. Okay, so say for example, we have this type of cross. So we cross this with a white parent. All right. So the resulting offspring will be the intermediate of the red and the white color, that is color pink. Ayan siya. So makikita natin dito na color pink yan siya. So because of the change in color in the phenotype, wherein it is observed to be at the median of the two extreme phenotypes, red and the phenotype white, we can see that this exhibits incomplete dominance. Okay, so hypothetically, kunwari, i-cross natin siya sa isa pang parent that is pink in nature. So ano yung magiging resulta ng cross natin? So this will produce the following traits. So it will produce a parent that is red, homozygous dominant, a parent that is pink, that is heterozygous, and an offspring that is white, that is homozygous recessive. So, genotypically speaking, ano yung ating genotypic ratio? 1:2 is to 1. Can you guess what is the phenotypic ratio, you guys? Phenotypic ratio. Remember, madi-distinguish natin yung red from pink from white. So therefore, the phenotypic ratio will be 1 red, eto siya, is to, all right, 2 pink is to 1 white. There you go. So makikita natin yung difference. Makikita natin yung difference in terms of the phenotypic ratios at the second generation. So 1:2:1. Okay? So are we clear? So that is for the case of incomplete dominance. Complete dominance, just one copy of the dominant allele is enough to mask the trait. Incomplete dominance means that the heterozygotes will exhibit the intermediate phenotype between the two parents. Okay, so guys, you might be confused with another type of interaction that is referred to as codominance. So this is the third type of allelic interaction. Codominance generally exists in the sense that kung titingnan mo, parang incomplete dominance siya. But instead of looking like the intermediate of the two extreme phenotypes, which is what we observe in incomplete dominance, in codominance, both traits will exist without interaction. Ano yung analogy natin dito? If incomplete dominance, it's like mixing one color of paint to another color of paint to produce a different color. In codominance, it's like mixing oil and soy sauce such that the two will coexist in the solution but will never mix. Okay? So anong common example natin dito? This is very commonly observed in coat color in cattle. So say for example, Coat color in cattle can either be brown. So let's say ito siya. C superscript B can either be brown or can either be, let's say, C superscript D, which is, let's say, dark red. Tapos, say for example, you have a parent that is containing this genotype. So yung kulay ng kanyang fur or coat will be brown. And at the same time, what if you have a parent with this genotype? So yung kanyang coat color will be dark red or close to maroon, as shown here. So the gametes produced will be the following. Oh, sorry. Red. Which will produce the offspring CB, CD. Ayan siya. Okay, so yung phenotype ng offspring na to, guys, will be that of a coat color that is comprised of both red and both brown. Okay? So makikita natin dito that the gene products of C superscript B and C superscript D will not exist. Or rather, will not interact. They will simply coexist. Unlike incomplete dominance na merong interaction and merong mixing ng gene products. Okay, so therefore, makikita din natin. So can you guess what happens if we cross this with another individual that is CBCD? Ano yung magiging phenotypic ratio? Can you guess? So the possible phenotypic ratio, so this is one parent. Okay. 1:2:1 pa rin, no? Or 2:1:1 pa rin. Correct. That's right. CBCB, CBCD, CBCD, CBCD. Here we can observe that your phenotypic ratio and your genotypic ratio will still be the same. Okay, so makikita natin that the coat color of this will be brown. Ito siya, brown. Ito siya will be roan. And ito siya will be the all right. Sorry. Mali lang yung nalagay ko dito. There. So 1:2:1 pa rin yung code color natin. Okay? So this is for the case of codominance. So ano yung discuss natin? We have incomplete dominance, we have complete dominance, and lastly, we have codominance. Let's now take a look at another very special type of interaction, and this is what we refer to as overdominance. Alam mo, overdominance pala. Actually, overdominance is very commonly used in breeding because the heterozygotes will have the tendency to outperform the homozygous. Ayan siya. So they have the tendency to outperform the homozygotes. So this has a very large importance in, let's say, agriculture and plant breeding. Hence, makikita natin dito na the term heterosis or hybrid vigor is very common. Because heterosis stage of overdominance, so that the heterozygotes will produce a more, let's say, a more high-performing phenotype. So say, for example, let's say you have a tomato plant. Tapos, kunwari, the gene Y yields for high yield. And its allele, small y, yields low. And you have two parents. Okay, so for example, a tomato plant sugar production. Okay, so let's say, for example, you have a sweet tomato plant here with this genotype and a sour tomato plant with not with this genotype here. So its gametes will be the following, and the crossing of the two will produce a heterozygote. And then, in the presence of the two different alleles that exhibit overdominance in the heterozygous state, this will produce a super sweet tomato plant, a tomato fruit. Ayan siya. So manotice nyo na the heterozygote will have a higher glucose production in its fruits as compared to its to one of the homozygous dominants, or rather to one of the homozygous phenotypes. So kung titingnan natin, can you guys guess ano yung magiging phenotypic ratio ng ganitong cross? Phenotypic ratio. So the phenotypic ratio will be 1 sweet is to 2 super sweet is to 1 sour tomato. All right, 1 is to 2 is to 1. And its genotypic ratio will be 1 is to 2 is to 1. All right. So I hope we're clear with overdominance. This time sa overdominance naman yung nangyayari is the heterozygotes will outperform the homozygotes, making their phenotypes very distinct and very different. Very important in crop breeding and plant breeding. Okay. So last 8 minutes for our last allelic interaction. Very interesting to siya. This is what you refer to as lethal genes. Yung lethal genes mo, this is the type of interaction wherein if you have a particular gene or a particular allele or a particular genotype, it will lead to pure lethality. So lethality means that you will be dead. So there are two types of lethality. First is dominant lethals, and another is recessive lethals. So anong example ng dominant lethal? Dominant lethals are lethal genes that if you have just the dominant allele, regardless if homozygous or heterozygous ka pa, but if you have the dominant allele, it will automatically lead to your demise. Common example is the Huntington's disease, which we have explored a while ago in your sample problem. Huntington's disease is coded by the big H allele, and then generally individuals with the Huntington's gene will only know of their disease once they reach 40. So late na nila malalaman na may Huntington's pala ako. And this will lead to a neural breakdown and eventually leading to one's death. So that's Huntington's disease that is carried by the dominant H allele. If you ask, bakit pa siya nagper-persist in the population if mamamatay din naman lahat ng mga individuals with the H allele? It is because of the fact that late yung onset niya. The individual would have produced offsprings before knowing that he is going to die at 40, thereby passing the trait on to the next generation. So lethal genes. Your recessive lethals naman are very specific in the sense that an individual has to be homozygous recessive for it to exhibit lethality. Whereas if homozygous dominant or heterozygote, mabubuhay pa rin siya. Have you guys encountered Manx cats? These are actually tailless cats. And these are cats that exhibit the Manx phenotype. Very special siya in the sense na, say for example, you have the allele S for spinal column formation. and you have S for no spine. Yung nangyayari dito is that if an individual with if an individual is homozygous dominant for the trait, he will have, or the cat will have a spine plus a tail. All right? But if a cat is heterozygote for the trait, he or she will still produce the spine, making him viable, but the cat is tailless. But if the individual is homozygous recessive for this particular individual, it will result to lethality because no spine will form and the baby will die at birth. So ayan siya. So yan yung Manx cat. So makikita natin dito recessive lethal versus dominant lethal genes. So ayun, na-discuss natin yung different types ng allelic interactions. As much as I want to, I'd like to discuss with you non-allelic interactions or epistasis, sana. Kaso it appears that we are a little bit pressed for time, but I'll be more than willing to provide you with instructional videos and lectures that can help you understand epistasis or non-allelic interaction. So, Sir Josh, do take it away. I think my time for day 1 is over.
Thank you very much, Sir Eman. There you have it, everyone. Assistant Professor Emmanuel T. Galang. Okay? So, dami pa natin napag-usapan ngayon, Sir Eman. No? And day 1 pa lang ito, meron pa po tayong 2 araw na kinompress ng ating buong course ng genetics, no? So,
Yes.
siguro, do we have any questions? Unay mo natin dito sa people in the Zoom. Meron ba? Ayan, sir. Mayroon pong tanong from Francisco.
Sir, is it possible to have genotype products of 25%, 30%, 15%, and 30%? Sorry, Francisco. Genotype products, you mean gene and genotype frequencies? Usually, because yung nangyayari dyan is, by virtue of meiosis, yung pagha-half ng ating chromosomes, or rather yung paghahati ng chromosomes natin will always be by a factor of 2. Palagi siyang pa-half ng pa-half ng pa-half ng pa-half. Although there are certain cases that might be exceptions to the rule. Say for example, if polygenic na yung trait, na hindi lang siya dalawa yung kine-carry na allele. Paminsan-minsan, there are traits kasi na hindi lang dalawa yung allele, so hindi lang pahati ng padalawa. And ma-observe natin yan siya sa ibang organisms such as plants and sa ibang traits such as plant height, such as other quantitative traits. Dito napapasok yung quantitative genetics, which we will be discussing in the future. But for the case of qualitative genetics, Sir Francisco, this is qualitative genetics. Na yung dinidiscuss natin is in the case of a gene pair. And yung hatian is on the basis of 2 lang, gawa ng meiosis. So, notice mo, always a factor of 2, always a factor of 2 if we're just concerning one gene pair. Ayan siya. But if, polygenic na yung trait, doon na siya pwede mag-iba.
Thank you, Sir Eman. How about all the others? Baka po may clarifications pa tayo regarding ang topic natin on heredity and chromosomal principles. Grabe, sir, from start to finish talagang tumutok sila ang ating mga 30 plus dito sa Zoom and 20 plus sa FB Live.
Thank you.
Mayroon pa ba? So I guess, wala na ba? Going once, twice, or siguro iipunin nila, sir, yung questions nila for tomorrow perhaps.
Yes, sir.
Yeah.
And also, you can email me at etgalang@up.edu.ph.
Ayan, sige pa,
In
sir.
case you have any questions. And estudyante ko yan si Jean and very active din yan sa class. Kaya andito din siya ngayon.
Thank you, Jean. Well, full support, no, kay Sir Eman. Okay, so I guess that's all for today. At this point, we would like to award the certificate of appreciation to our distinguished resource speaker this morning. So allow me to read the citation. University of the Philippines Los Baños Learning Resource Center presents this certificate of appreciation to Emmanuel P. Galang for serving as resource speaker during the GENE-ius Talaga tutorial webinar series on genetics, session 1, entitled Chromosome, Heredity and Chromosomal Principles, held today, December 6, 2022, at the Learning Resource Center, UPLB College Laguna, signed by our Director, Assistant Professor John Mervin L. Embate. Thank you very much, Sir Eman.
Thank you. Thank you din, sir, for the invitation. I look forward to our meeting again tomorrow. More interesting
Yes, sir.
topics coming our way, mutations and DNA and all that.
Yes, sir. Thank you very much. All right. Now for the evaluation of this session, please do not forget to answer the evaluation form after our Zoom meeting today. Okay. So the deadline of responses is 10 p.m. today. So we highly appreciate your comments and suggestions on how we can further improve our services at the Learning Resource Center. So the evaluation link is being shown right now on your screen. And let me place that also in the comment section so that you will be directed immediately to the evaluation form. There you go. Okay.
Sir, padagdag na lang din po ng email if in case
Yes.
manghingi
And
sila ng lecture videos ko po.
okay po.
etgalang@up.edu.ph. There.
Yes. So if you have queries or concerns to Sir Eman, he provided his email address, etgalang@up.edu.ph. Okay? You may also scan the QR code so it will be directed. All right. Again, please be reminded that the deadline of responses is only until 10 p.m. tonight. So that's tinyurl.com/GENE-ius, capital GNE, eval session 1. All right? Okay. So we hope that we gain all inspirations and valuable insights from our speaker. So reminders for our next session, previous slide, please. So our next session is tomorrow, December 7. So ang title naman natin is You're My Genotype. So how genes function within. So it's another set of topics on genetics that we'll be discussing tomorrow, December 7, Wednesday, same time, 9 a.m., still with Sir Eman. Okay? So at this point, we would like to recognize and thank our team, #Team LRC, and our GENE-ius Talaga team, headed by our director, Sir J.M. Embate. On our technical side is our student assistant, Ms. Sofia Gabronino, and our team here at LRC, Ate She, Ate Majeng, Tita Allen, and Kuya Ewak. And a big thank you again to Sir Eman, our very passionate speaker, for this morning. So thank you so much, everyone, for joining us. Stay hopeful, stay healthy, and stay safe. We'll see you again tomorrow. Okay, thank you.
Maraming
Oh,
salamat.
before we leave, let's first have a group picture
Yay.
since we're only a few here. Okay, may I request those who can turn on their cameras to turn on their cameras.
Ay, naka-disable ata, sir.
Ay,
yung
wait,
camera.
let me allow para. Sorry. Wait. Let me enable everyone to turn on their cameras. Ayan. I think pwede na. Ayan. Okay. Enabled na po.
It's nice to see fellow educators here as well. If you need help with any topics or visual aids and stuff, marami akong drawings. Hindi kagandahan pero baka makatulong.
All right. Is everyone turn on their cam? Okay. Sige. Let's have our photo taken. All right. Ready? All right. Ready. 1, 2, 3. Smile. Okay. And next panel. 1, 2, 3. Smile. Okay. Thank you very much, everyone. And we'll see you tomorrow. Okay.